Hello and welcome to our presentation on electron microscopy. During this presentation we'll talk to you a bit about an overview of what electron microscopy is, what is it, etc. Then we'll go more in depth into the two different types of electron microscopy, showing their advantages and disadvantages, and then right at the end we'll be talking to you about data analysis, how to deduce the data that we have collected from this methods. So to start off with, I thought we'd look at a quick overview of electron microscopy, just so when everything goes a bit more in depth, we'd have a bit more idea of what's going on. So to start off with, electron microscopy. It uses steady beams of electrons as a source of illuminating radiation. These produce high-resolution images of the sample, which could be structural bases of the cell and function, or it could also be shown to show certain diseases, viruses, etc. that are on the sample. Combined with other methods such as thin sectioning, immune lab labeling, negative staining, these are more specific methods to get more specific answers. For example, we can use the negative staining to highlight certain parts of the cell that we would like to focus on, and etc. with the other ones. Now there's two types of electron microscopy. There's transmission and there's scanning, and in this presentation we'll be taking you through both methods and their advantages and disadvantages.
What is scanning electron microscopy, or SEM? Scanning electron microscopy can predict the surface of a solid and produce 3D images of the specimen analysed. This analytical technique happens under vacuum. This reduces the number of electrons hitting the surface of the specimen, so they can be detected. Also, it reduces the interference from outside sources. How does it work? From the scanning electron microscope diagram shown, electrons are released from the electron gun with the beam containing high energy electrons with an energy between 2 to 1000 kilo electron volts. The electron beam becomes focused as it passes through an anode and magnetic lens. The beam of electrons is scanned over the specimen, and high-energy backscattered electrons are reflected off the surface of the specimen. These backscattered electrons are then detected by a secondary detector, and electrons and data collected are transformed into images. In scanning electron microscopy, these images can have a resolution between 1 micrometer and 1 nanometer, depending on how focused the incident beam is, how it's scanned across the specimen, and how much the beam spreads out through the sample before it is reflected.
Now that we know a little bit about SEM, we can talk about its advantages and disadvantages. Its first advantage is 3D images. For example, like the image of starch, we can see these images in 3D, which can provide us a little bit more analysis. A second advantage is that we can improve on its disadvantages. For example, by using other tools with SEM, such as an optical microscope, we can help improve signal-to-noise ratio. A final advantage is that we can change the electron source. When changing the electron source, we can use electrons such as lanthanum hexaboride electrons, which are good at low accelerating voltages and can provide brighter images. Now we can discuss the disadvantages of SEM. As you can see, SEM can be quite a large bit of equipment, which means it has to be kept in its own room away from vibrational and magnetic interference. SEM also have highly specific equipment that can cost quite a lot of money. SEM also has issues with repeatability. Due to SEM taking longer measurement scans, measurements can appear less repeatable. This happens when there is more variability within a sample measurement, which means every sample will slightly differ in multiple screenings. Finally, with some SEM, we can see specimen damage. At high accelerating voltages, radiation damage can be observed. On this slide, we'll be talking about transmission electron microscopes, also known as TEM. TEM uses various different techniques. Most use electron holography. Measurements are done when we measure phase changes of electron waves when it passes through the sample and compare them to when it passes through a vacuum. Images of TEM are monochromatic, which means it comes only in black and white. We make images visible by electrons hitting a fluorescent screen or by digitally capturing them. Here you can see a photo of a TEM. TEMs are useful for looking at thin samples, so we can detect defects in semiconductors, or we can look at cells, such like the cell wall. Here are the references used for this slide.
And finally, we have come to the end of the presentation with data collection, how we actually take everything we've learned to apply these methods to gain some sort of data. So as you can see here, we have our two different examples of electron microscopy. We have the TEM and the SEM. We'll look at the SEM first. As previously discussed, SEM is a focused beam of high-energy electrons to generate a variety of signals at the surface of a solid specimen. Now, these signals can be produced by secondary electrons, backscattered electrons, diffracted backscattered electrons, photons, visible light, and heat. These are all things which can be used to generate this image right here. Now, with this image, we'll also need a previous understanding of certain structures, because this could only get us so far. We have the image, but then we'll also have to deduce it, because, for example, secondary electrons, they produce the image itself. We have the backscattered electrons, the diffracted backscattered electrons. These things are used to deduce the orientation and the crystal structure of the specimen, of what we're trying to look at a much closer angle. So then we can see the actual structure. We can see the crystal structure, or the small spikes over here, but it's not as detailed as the TEM, which obviously, as we previously discussed, is a high beam of electrons through a thin layer of sample. Now, these can give us more detailed images. As you can see, we can see the photo of the cell. We have the cell nucleus for here. We can see the thin line of the membrane on the outside. And this could also be used, for example, looking at different transitional phases, for example, metaphase, anaphase, telophase, all of those good stuff. And then also, like I was mentioning about diseases, we have an example of a virus. Now, the reason we're able to tell this is a virus, because we can see its structure. We've got the protein. We've got the protein shell at the top. We've got its tails at the bottom. And this is how we obviously deduce the images. We'll need a previous understanding of what we're actually looking at, just so then we'll be able to give these structures names. For example, we have the crystalline structure of here. We see the small spikes of the asbestos. Obviously, we all know asbestos is bad for you. And then over here we have the cell with we're able to give all the different parts of the cell its names. Same with the virus. In conclusion, during this presentation, we discussed what is electron microscopy, a bit of an overview. Then we looked more in depth into TEM and SEM, with looking at advantages and disadvantages of both. And we also looked at data collection, different results and how to analyze them, and how they might require a bit of previous knowledge in biochemistry to understand some of the organic samples. Thank you so much for listening, and I hope you enjoyed.